Today, we're really privileged to have Dr. Jordan Marrocco, who will be talking to us about the influence of sex in genomics, epigenomics, and hormones of allostatic load. Before I introduce him, I just want to acknowledge that we live, work, play, and participate in a community on the unceded, ceded, and traditional territories of the 630 First Nations, each of which possess their own unique traditions and history here on this land that we now refer to as Canada. We acknowledge the importance of the Truth and Reconciliation Commission of Canada's call to action and the United Nations Declaration on the Rights of Indigenous Peoples. In all of our work, we're committed to ensuring Indigenous women's rights to health and safety and the equal opportunity to participate in a manner that recognizes and respects Indigenous cultures and traditions. Today, I'm joining you from Toronto, which is covered by Toronto Purchase number 13, with the Mississaugas of the Credit. Toronto is now home to a vast diversity of First Nations, Inuit, and Métis who enrich the city. Many of our members also live in Vancouver, which is part of the unceded homelands of the Coast Salish peoples and the traditional territories of the Musqueam, Squamish, and Tsleil-Waututh First Nations. The Women's Health, this is brought to you by the Women's Health Research Cluster. We're growing all the time. We have over 750 members across 32 countries. It's free to join. Please tell people and join yourself. We have some awards, trainee travel awards that you can apply to, but you have to be a member, so that's how we get you. We have a number of events, research trainee research presentation series, this series you're in right now, some community events, some conferences. The past ones are on our YouTube site, and we have workshops as well. We are just very pleased to bring you evidence-based content on women's health, including these seminar series. So I'm going to stop sharing. And while I stop sharing, Dr. Marrocco can upload his slides or share his slides, and I will read out his bio. Dr. Marrocco is an assistant professor of biology and psychology at Touro University in New York, and he's also an adjunct assistant professor of research in neuroscience at Cornell. His research focuses on the epigenetic signatures of stress and steroid hormones at different developmental periods in preclinical models. Over the last 15 years, he's used behavioral, pharmacological, and genomic methods to demonstrate the molecular mechanisms of stress-induced allostatic load and its association with increased risk for psychiatric disorders. Dr. Marrocco received his bachelor's degree in biological sciences and a master's degree in neurobiology from Sapienza, something like that. Sapienza, of course it is, University of Rome, and a Ph.D. in neuroscience from the University of Lille. During his postdoctoral training, he worked under the mentorship of the late, great, amazing Dr. Bruce McEwen. We both did a postdoc with Dr. Bruce McEwen, so that's why we know each other. And if you ever had the fortune to know him, you know he was just a really amazing human inside and out and did some really great science. Dr. Marrocco not only does amazing science, but he is a member of the Diversity, Equity, and Inclusion and Education Committees of several scientific societies, including the one for Women's Health Research Cluster. I don't know if he still is, but he was, but also the Organization for the Study of Sex Differences, ALBA, and ACP. And he is really a true champion for equity, diversity, inclusion, and accessibility. So we all thank him for his service in that area, because it's a long hill, and a hill we continue to climb to strive for greater inclusion. Now, you know that I always ask for a fun fact. The fun fact here is that he has almost exclusively been wearing custom Converse shoes that he designed for the past 20 years. That's incredible. Now I really need to, I've never noticed your custom Converse shoes, so now I need to notice them. And he is a very good singer. He speaks fluently and sings in 4 different languages, but his caveat is he only sings at conference karaoke. So I think we need something so that we can hear him, right, crowd? Without further ado, please take it away.
Thank you so much, Lisa, for the great introduction. It was flattering and fantastic. And thank you so much for inviting me and for having me in this incredible and important initiative organized by the Women's Health Research Cluster. So I'm excited to talk to you today about the influence of sex in genomics, epigenomics, and hormones and allostatic load. So let me start first to know how why it is important to study allostatic load, and with this cartoon that was drawn by Bruce McEwen in the late 90s, which shows what we called an allostatic clock. An allostatic clock meaning that in fact, whenever we have an experience in life, there would be environmental stressors or major life events or trauma and abuse, but put on top of this, any experience that we have, they are perceived by the brain as a threat or not threat. They would induce behavioral responses. As we know, behavioral responses can be fight or flight or other personal behavior. And these perceived stress events are also influenced by individual differences. Individual differences can go genes, so included sex, for example, development and experience. And this system induces physiological responses of adaptation, which we call initially allostasis. So in allostatic adaptation, meaning that there is a new degree of adaptation to these perceived stress responses. But there is also an accumulation of these allostatic responses over time, meaning that we, in fact, tend to accumulate, and whatever we experience, we would accumulate this experience in a sort of clock that always goes forward. So we cannot really clock roll the clock back in sense that whatever experience do we have that accumulate over our life. Is this a positive thing for adaptation? Yes, of course it is, but sometimes it is not. In fact, allostatic load, since it is promoted by cumulative biological burden that is exerted on the body through attempts to adapt to life's demand, it is promoted with the same mediators that allow us to adapt, but they can also cause damage if they are overused and out of balance. So the question is, where is the limit in which we are in sort of moving through this adaptation physiology and system? So we have an adaptation window in which we can move. So the question that I will try to respond, and specifically how this question can be important in studying sex differences and also women's health, is understanding where the biology limits our responses and our responses to the environment. To start understanding the response to stress, I always like to point out that the response to stress is biphasic, meaning that there is also a good stress and it's always a bad stress. So there is a good stress, which, for example, is what here is highlighted in blue. And it's a stress that can be an adaptive stress, which levels of, in terms of adaptation in the brain, let's say, we can give some examples such as enhancement of synaptic function, transmission, long-term potentiation. And it depends, of course, the degree of which the stress is good or toxic stress. It may depend on the time, so the duration of stress, but also on the intensity. So let's say that the stress is more intense, or it's longer than short stress needed for adaptation. Then at that time, with the increased amount of frequency of stress, we can look at this second part of the descending part of this inverted U curve, in which we do fall into what we call allostatic overload. Allostatic overload in this second part of the curve mostly is associated with maladaptation. And I have some examples on the right here. For example, loss of synaptic function, loss of adaptive plasticity, resilience, damage of potentiation, and sometimes may also induce a mechanism of early aging of the brain. To study stress, I'm sure many of you are aware of with this chart. I put this chart because we are mostly aware of the hypothalamic pituitary adrenal axis and how it is stimulated by stress. But I want to highlight here a structure that is important in controlling the hypothalamus and also receives stress responses also receive stress input because of high concentration of glucocorticoid receptors, for example. And this the structure that I will focus mostly on my talk about is the hippocampus. The hippocampus has been the center of my research attention. So it's the region I like the most. So take it in a way of it's a love connection with the hippocampus, but also I will try to convince you why the hippocampus is important in the response to stress. When we talk about the hippocampus, I like to distinguish the neurostructure, neuroanatomy of the hippocampus and say that it is important that the dorsal and ventral hippocampus are considered as functionally distinct brain regions because of their unique anatomical connectivity, but also because recent evidence shows that in these subregions of the hippocampus, there are discrete set of genes that really characterize and distinguish these two brain regions. Of course, the study that I'm showing, that we show you today, are mostly focused on rodent brains, especially in mice, in which I would distinguish, in fact, both the neuroanatomy, the gross neuroanatomy of the hippocampus, the dorsal and ventral hippocampus, but also the subfields of the hippocampus, especially the CA3 regions of the hippocampus, which is characterized by CA3 pyramidal neurons. You can see the CA3 region of the hippocampus here, which receives connection, receives muscle fibers from the dentate gyrus, and CA3 regions receives also fibers from entorhinal cortex, and in turns through the Schaffer collateral fibers connects and projects to the CA1 pyramidal cells. The CA3 regions is also an important nexus of information processing for spatial and episodic memory. It contributes to the negative feedback in the regulation of the HPA axis, and it is an important neuropsych of structural plasticity. Cannot not mention the CA3 region and how important it is in sex differences without mentioning this pillar, seminal paper by Lisa Galea that shows, in fact, that CA3 region is an important region for anatomical plasticity and sex differences. This is an example on how chronic stress, in fact, induces shrinkage in the apical dendrites in males, but shrinkage in the basal dendrites of females. So we understand that some regions are particularly important to study sex differences and specificity of the action of stress. To go a bit in depth on what I will show you today, and I will try to decompose my talk. I will show you today a different way and different combinations on which we can study stress and we can understand stress response. I will use, and as Lisa also introduced, as I've used through my research and my research philosophy to approach to approach stress research and to answer some of the questions. Environmental models, which consists in forced stress or acute restraint stress, or even environmental enrichment in a collaboration that I will show you at the end of my talk. Hormonal challenging, or corticosteroid or estradiol, but also genetic modifications. In particular, I will show you like a single nucleotide polymorphism called BDNF Val66 MET. It is important when, particularly when we study women's health. Let me start with this. Let me start with the interest that we had on this interesting single nucleotide polymorphism and how these interact with estradiol. This single nucleotide polymorphism it's BDNF polymorphism that consists in the substitution of valine with a methionine in position 66 of the BDNF. And it's a variant that is prevalent worldwide that modulates psychiatric disorders. And you see that prevalence, in fact, is strangely differs across ethnicity, if we can call it this, but also across geographical locations. So some geographical location, the prevalence of this SNP is higher than others. And one would question, why is this SNP so conserved across the world population? Is this an advantage or a disadvantage somehow? And that's what's important sometimes to ask question of the persistence of some conserved domains, and do we say is this domain conserved across species, or is this domain, or is this something that only belongs to humans? To respond to this question, in fact, there are some papers from this NIMH group led by Karen Berman, who showed that brain-derived neurotrophic factor genotype, the Val66 MET genotype, and ovarian steroids interactively modulate working memory-related hippocampal function in women. Say that the activation here of the right hippocampus when Val, which means wild-type women and MET carriers are under estradiol, is different when they are treated with Lupron, that in fact blocks the menstruation. There are no more differences. But then when progesterone is given back, there are it really depends on the modulation of working memory. And this paper shows that it's typical, the modulation of the more working memory in VALMET carriers is specific of estradiol. Moved on that, we decided to work on to mimic this idea of the intersection of VALMET with hormones with using a model of BDNF Val66 MET polymorphism in collaboration with Francis Lee Cornell University that has engineered this model in mice in which, as I mentioned earlier, methionine, valine is substituted with a methionine in position 66. This model has been shown to impair BDNF signaling mediated by the ligand. So when I say that, it means that it's not the amount of BDNF that is in fact impaired, but rather the signaling of BDNF. It also disrupts the intracellular trafficking of BDNF, affects activity-dependent release of BDNF, modulates the neurotransmitter release and long-term potentiation. And interestingly, BDNF heterozygous VALMET mice, male mice, show increased HPA axis activity as well as increased affective behavior, but only after subchronic stress, and not female mice. So using this model, we asked, what is the genomic response in female VALMET mice to oral estradiol? Mice were overreactomized and then left to recover and then treated with 300 nanomolar of beta estradiol in drinking water for 2 weeks, then were tested in open field, a splash test, and then the ventral hippocampus was isolated for RNA sequencing. What you see here is a z-score of a combination of these two tests when we combine all the behavioral variables in a z-score by highlighting what is an emotionality score, meaning that the variable of high emotionality have a plus sign and the low emotionality have a minus sign. And when we combine the open field variables and the splash variable, we found that estradiol at back in overreactomized mice induces behavioral ability, but only in BDNF Val66 MET mice. Now, you may remember this article that I showed on the behavioral ability on BDNF Val66 MET women on cognitive approach in this test. But in fact, not only that, but the behavioral ability that we do see under estradiol somehow may recapitulate a behavioral phenotype that is observed in women with premenstrual dysphoric disorder, which luperon, in fact, somehow reduces the negative symptoms associated with premenstrual dysphoric disorder. But then when women are given back progesterone and estradiol, these symptoms come back. And in collaboration, because of this phenotype, we were curious to see how this would intersect translationally with human data. And we collaborated with Peter Schmitz at the NIMH and found that there was a transcriptional overlap by doing a sequencing of the ventral hippocampus in mice and lymphoblastoid cell lines in humans. We found that there was a transcriptional overlap of the genes found in the ventral hippocampus from mice and from women with premenstrual dysphoric disorder. You see here that E2, so estradiol-regulated gene, when we overlapped women and mice. So careful that this is in fact cross-species, cross-tissue, because for the women, we had a sequencing in a lymphoblastoid cell line, and in Valmet mice, we had the ventral hippocampus, so it's both cross-species and cross-tissue. We found conserved estradiol-regulated genes. And then we asked ourselves, what are these conserved epigenetics, so these conserved estradiol-regulated genes? By composing a map of connectome of these estradiol-regulated genes in human and mice, we did find, in fact, that were clusters of epigenetic modifiers that were common in women with premenstrual dysphoric disorder and Valmet mice. And interesting, this complex that was found to be common across species was an epigenetic complex, which is a multimeric protein complex that methylates lysine 27 and lysine 9 residue of histone H3. Meaning that, in fact, there is a direct control of gene expression mediated by estradiol that may show a common behavioral phenotype and, in fact, is conserved across species. That's basically what I have summarized here in this slide. So estradiol add back, specific behavior, emotional ability in Erecto mice, BDNF-Val6 MET mice, epigenomic intersection of BDNF-Val6 MET mice with women with premenstrual dysphoric disorder under estradiol, and conservation of this epigenome under similar hormonal condition. We take one step forward using a hormone that is dear to many people studying stress, which is corticosterone, which is the cortisol. It's called cortisol. Is prevalent in human. Corticosterone is prevalent in rodents. It comes from the same chain of biosynthesis of steroids, basically. So we, on one question that we wanted to answer here is how corticosterone, in fact, intersects with this SNP, with the BDNF-Val6 MET, given that, like estradiol, corticosterone also is a steroid hormone. We use this model that is that consists in drinking water low dose oral corticosterone. It's a model that was established in the McEwen lab by Ilya Karatsureus in the beginning of the 2000s. And the advantage of this model, you see here in the x-axis, white and black ranges. And white here is the light period and the black is the dark period. Yes, you do see that there are high levels of corticosterone when the animals drink in the nighttime. But you do also see that what is important is this model is that the rhythmicity of the corticosterone is somehow maintained. So the circadian cycle of corticosterone is not disrupted. So when we would do behaviors in the afternoon, for example, we would have a normalization of level of corticosterone. So what we would see, in fact, is an alteration that is not dependent on the time of the behavioral observation, but rather on an effect that occurs across time. The low dose of oral corticosterone, one of the main effects is also disruption of the HPA axis activity, which induces behavioral susceptibility. You see here the level of corticosterone measured a basal level in vehicle treated mice and cohort treated mice, and levels of corticosterone after stress in vehicle treated mice that mount a good response to stress, in which in corticosterone treated mice, the response to stress is blunted, indicated that, in fact, the HPA axis activity and response to stress is impaired in corticosterone treated mice. We asked ourselves, well, what happens if we use this model in males and female mice, and particularly also if we include this SNP mutation? the BDNF Valmet, and mice were treated with low doses of oral corticosteroid with 25 milligram per liter. Then after 3 weeks, they were tested in the light-dark box, then in the splash test, and then the ventral and dorsal hippocampus were both isolated and processed for RNA sequencing. The main phenotype, once again, what I'm showing here is an emotionality score, which is a score that combines different variables of these two behaviors, in which positive score means higher emotionality and negative score, lower emotionality, shows that both in males, corticosteroid induces increased emotionality score, which means increased anxiety-like behavior and depression-like behavior, regardless of genotype. You see that the wild-type mice, male mice treated with corticosteroid, the emotionality score is higher, and with Valmet mice treated with corticosteroid, the emotionality score is higher. In female, interestingly, there were no changes in wild-type females, while in Valmet females, there was an opposite phenotype. Now, this does not mean that since their emotionality score is lower, it means that they were less anxious, because these tests are built and validated to measure anxiety rather than not anxiety. But it does mean that there is an opposite behavioral effect and an opposite phenotype observed in males and females, only and only if the BDNF Valmet SNP is present. How this showed and correlated with the dorsal and ventral hippocampus, specifically the ventral hippocampus that regulates affective behavior, where what I show here is a rank-rank hypergeometric overlap, which means basically comparing two data sets, and the two data sets of comparisons are one on the y-axis and the one on the x-axis, and understanding if there is a genome or gene expression concordance or discordance. This on the right summarizes what concordant and discordant gene expression means. And basically in these two quadrants, so the lower left and the upper right, we would have an increased heat map. The heat, so basically the color, correlates with how many genes are concordant or discordant. On the bottom left, there are genes that are in both data sets that are indicated on the X and Y axis, downregulated. On the top right, genes that in both data sets are upregulated. And in the other two quadrants, the genes are discordant. So if in one data set is upregulated, in the other one is downregulated, and vice versa. By looking at the dorsal and ventral hippocampus and comparing the data set of Valmet mice and wild-type mice in females and males, and the ventral hippocampus in females and males, we do see a particularly interesting genome overlap similarities. In fact, this rank-rank epigenetic overlap show that court-induced differentially expressed genes in BDNF Valmet females were discordant for court differentially expressed genes in wild-type females. Once again, highlighting this particular genotype, the expression of BDNF Val6met females, which recalls this unique behavior that we did find in Valmet females, but not in wild-type females. When we then compared this time males and females instead of wild-type and Valmet, we do find that rank-rank hypergeometric overlap shows that court differentially expressed genes in the hippocampus of Valmet males were discordant from court-induced genes in the hippocampus of Valmet females. Well, indicating in fact, if you want to highlight here, this is the discordance in the ventral hippocampus, that in fact the specificity of this behavioral phenotype that we did see in Valmet females also is related to this discordant gene expression in the ventral hippocampus. We then told ourselves if these may be related to specific clusters of genes that regulate the response to stress, and especially genes that participate in the signaling of GR. And in this case, we isolated and focused on GR binding genes, but also epigenetic modifiers. And we did find that when we cluster the groups in the dorsal hippocampus, the groups would cluster by genotype. So the wild type would cluster together independent in terms of sex, and the Valmet mice would cluster together, meaning that their genomic distance was mostly influenced by genotype rather than sex. While in the ventral hippocampus, the opposite was true. Their genomic distance was in fact influenced by sex, but not by genotype. We would see that, in fact, when we clustered these genes, experimental groups clustered by genotype, so Valmet and wild-type males clustered together, and wild-type and Valmet females clustered together, meaning that in the ventral hippocampus, there is a strong genomic drive for sex differences rather than genotype. By doing a methylated DNA immunoprecipitation, because we wanted to see what drives in the genomic of these specific genomics of sex differences based on the GR binding genes, we did find that the glucocorticoid receptor gene 1, so NR3C1, GR1, was specifically differentially methylated and specifically on exon 1 and F in the ventral hippocampus of BDNF multisystem females, which brings together on the specificity of this behavioral phenotype that was specifically selectively observed in Valmet females. In summary, what we did see is that corticosteroid caused increased anxiety and depression-like behavior, so emotionality score in males, but not in females, and specifically females show a reduction, so an opposite phenotype behaviorally after port treatment that was associated with increased methylation of specific promoters of the NR3C1 gene. So the next step question is, but is this happening even if we give environmental stimuli? And we use this model of forced swim stress to analyze how was the response to forced swim stress if we would challenge male and female mice that were both wild type of BDNF Valmet. After 40 minutes recovery, after 6 minutes swimming, the mice were then sacrificed and the hippocampus was collected. This time, I want to focus on a unique highlight that concerns the study, which was the isolation of the CA3 pyramidal neurons. As I showed you at the beginning of my talk, CA3 pyramidal neurons are very important for the response to stress. And to do that, we used this model called Giprin3 backtrap. So we used the backtrap technology in which we could isolate specifically CA3 neurons by using these methods of translating ribosome affinity purification. You see here the enrichment of the trapped region that was immunoprecipitated out of the hippocampus, and you see that the trapped region, in fact, has this marker that indicates that the regions that we had isolated was specifically a region, the CA3 pyramidal neurons. Interestingly, when we did RNA sequencing on this homogeneous population of CA3 pyramidal neurons, we found that acute stress, when we compare females and males, activated discrete translatomic because now I call it translatomic because we had isolated the ribosome. So what we isolate is basically the translatable part of the gene expression. And there was an important sex differences in males and females. And as you can notice, in fact, females had higher reactivity in terms of gene expression compared to males. We also compared males and females to their matched the wild type males and females to the matched BDNF Valmet females that were not stressed. And once again, we found that BDNF Valmet females, but also in this time BDNF well-met males, they show a phenotype of stress even in the absence of stress. We had seen that this was true for some of the previous genotype evidence that I shared with you. But this was incredible because, in fact, the differential expression gene similarities between wild-type mice that were subjected to stress and well-met mice that were not subjected to stress was really overlapping, indicated sort of pre-stressed phenotype, a pre-stressed genotype in this case, of BDNF well-met mice. In fact, so a summary is that force-swim stress induces a genomic response which is different in males and females, which is higher in females compared to males, and that it's similar to the genomic response that we do see in genomic response, but more like a genomic landscape that BDNF well-met mice do have regardless of being exposed to stress or not. More recently, we have adapted this model of force-swim tests to understand why we would have these differences in gene expression, and studied, in fact, a combination of force-swim stress model with acute restraint stress to understand whether one experience of force-swim stress could last over time and somehow influence a second exposure to a second stressor. To do this, we first stressed the mice in acute restraint stress, and then tested the mice in light dark box. I'm going to say I'm not showing these results because we did not find any differences after the week and after one single episode of acute restraint stress. And the day after the behavior, the mice underwent force-swim stress as a second stressor, so as a second hit stress. And then the ventral hippocampus was collected and processed for attack sequencing. I'm going to go in there. So why attack sequencing? Attack sequencing because at this point, we were interested in the regulation of gene expression. So when I say regulation of gene expression, what we are mostly interested in is what are these mechanisms that influence what genes can be expressed or what genes cannot be expressed. And to do that, we did a sequencing on the actual chromatin to understand what sections of chromatins were open, so they would somehow favor the expression of that particular gene, or what sections of chromatin were closed, so they would in fact prevent or reduce the expression of a certain type of gene. And this is the pipeline on how the attack sequencing of this assay for transpose accessible chromatin works. Basically, it is a tagging of transposons that would tag pieces of DNA depending on whether the chromatin is open or closed, depending so where the genes are more or less expressed. And then they are fragmented and amplified and sequenced. And the results that we would have is basically this section of DNA is a section that is more open than another section of DNA. And the question that we ask is, so if this section of DNA is more open, what is the section of DNA, in fact, sequencing or codifying for? Here you have all the experimental groups that we analyzed. You have control mice, females and males, mice that were exposed to only one episode of acute restraint stress, mice that were exposed to only one episode of force-swim stress, and mice that were exposed to both stressors with 1 week apart. And across the most significant altered chromatin regions across these groups, we found that the androgen receptor, mineralocorticoid receptor, and glucocorticoid receptors were the most significant altered chromatin regions. So these regions binding for these three important important actors of the stress response. We did find that the epigenetic effects of acute restrained stress, if you see this, this is a heat map basically in which the blue indicates that the chromatin is close, is more close, and the yellow indicates that the chromatin is open as compared to the other groups. What we found is that the epigenetic effects of acute restrained stress persisted 1 week after stress and changed chromatin accessibility for the binding site of androgen receptor, mineralocorticoid receptor, and the glucocorticoid receptor. You can see this, in fact, by seeing how the color changes if the mice are exposed to both stresses as opposed to one stress, indicating that the previous experience of acute restrained stress somehow would influence chromatin opening. And this is basically what also I have summarized here. So one single episode, in fact, can be carried 7 days later and influence the second episode because some regions of the chromatin do remain open and their accessibility changes because of one individual experience. This is quickly summarized here. We did see sex differences. We did see that sex differences did persist even after one individual episode of acute stress, whether it is acute restrained stress or full swing stress, and that sex differences also persisted after a two-hit stress, and that this was associated with specific regions of the DNA that would respond to and somehow prepare the individual, or in this case, the subject, to a second response to stress. Once again, how epigenetics, in fact, has shaped and prepared this particular region of the brain that was eventually hippocampus to a second response to stress in terms of DNA reorganization. To conclude, let me go through this last part, which is a collaboration that I carried with Dr. Kettner and Massachusetts College of Pharmacy and Health Sciences, in which we introduced this idea of understanding what environmental enrichment would induce to the genomic expression. But what is particular in this collaboration is that what I'm going to show you is, rather than the genomic expression in the brain, what we did, in fact, is studying the genomic expression in the milk. So mice were arrived in the animal facility, and they were immediately assigned to standard or environmentally enriched housing. At birth, then there were other standardized litters or cross-foster litter. Then the milk was collected postnatal day 10, and an open field, a social behavior open field, was conducted at postnatal day 21. Behaviorally, we showed that postnatal environmental enrichment was associated with greater sociability in males, while prenatal environmental enrichment was associated with greater sociability in females. And what about the milk? Well, in the milk, we did a transcriptomic analysis of P10 milk, postnatal 10 milk, from mothers from dams living in environmental enrichment or standard housing. And here it's a volcano plot showing the genes that are significant across the groups. And we did target some important pathways in milk content, namely what you see here: triglycerides, nutrient oxytocin, GR signaling, epigenetic, and GR binding genes. Overall, when you see like these heat maps represents what genes are upregulated in yellow and downregulated in blue, you would see that when we compare standard housed and environmentally enriched dams, milk from dams, you would see that for many of the genes that we identified, there is opposite gene expression and gene regulation. meaning that, in fact, the experience postnatally and prenatally plus postnatally environmental enrichment can influence gene expression in the milk, and this induces differences in the behavior that result in sex differences in terms of behavioral sociability. Overall, I always like to conclude with the idea of the inverted curve that I started with, adding to this inverted curve what I hope I could show you today, which is an integration of adaptation, time intensity, and frequency that would influence allostatic load with a third variable with a z-axis that can be moved and can adapt and mold and shape this curve and the shape of this curve. And this axis, what pulls the variables that could pull our response to stress, and this allostatic curve can fall into genetics, hormones, and environmental stimuli. With this, I want, of course, to thank, first of all, Bruce McEwen, which was the late Bruce McEwen, which was a beacon for my research, for most part of this work, which I miss very much every day. And then all my collaborators at the Rockefeller University, at Cornell University, at NIMH, at Massachusetts College of Pharmacy, and the University of Illinois at Chicago, and Touro University for keeping me working on more of these interesting experiments today. And thank you, everyone, for your attention. I'm here for questions.
Well, thank you so much, Dr. Marrocco. That was excellent. What a tour de force from, opening and closing histone modifications to environmental enrichment to lactation, or at least an analysis of milk. That was really amazing. That I have a few questions, but I see that there's lots of little comments coming through.
Would you like me to stop sharing, or shall I keep?
You can keep it. It's up to you, whatever you want. For those of you, so just so you know, which you probably can see, but you have a number of thank yous. But for the rest of you, if you can do some emojis, give a little clap, because we're virtual. It's always nice to see them. I have a couple of questions, but also if you have a question, please do put it in the chat, or you can unmute yourself and ask your question as well. And I will maybe kick off if nobody's raising their hand yet. Sometimes it's hard for me to look at all the emojis, but I have a few questions, and mostly about the histone study that you did, the chromatin opening, closing. Sorry, I can't speak because I have to get on a flight, so I'm not thinking clearly. I think it was CA3, right? You did it in the CA3 region?
No, the chromatin, sorry, it was ventral hippocampus.
Oh, ventral hippocampus. Ventral hippocampus. Okay, so I might have. Okay. So in the ventral hippocampus, and you saw that the three, was it glucocorticoid, mineralocorticoid, and androgen receptors? Why not estrogen receptors?
No, in fact, these were not picked.
Oh, it wasn't picked. Okay.
That's where that's a great question. Those were, if we did analyze across all genes.
Yeah.
Then bioinformatically, there is a prediction of what are the region of the DNA that are mostly differentially opened across all groups.
Okay.
In fact, these were these regions that then we did a prediction on what are the genes that, or the receptors, in fact, that bind that case these regions. And this particular sequence was associated with RGR and AR.
I see.
But we did not find the estrogen receptor was
particularly high, at least.
Yes, it was particularly differentially.
Or different, sorry,
yeah. But it's also, there is a bias, and thank you for giving me this opportunity to explain this. It's because it really depends on what we are comparing across groups. It is likely that if we go back, and it's something that I'm planning and I'm kind of doing is that if we do focus on females only, because
Right.
studies when we focus on females only, if we focus on females only, do we have a different outcomes in terms of those opening parts of chromosomes?
Oh, I see what you're saying. Because does the algorithm or does the statistical method it groups everything and says, okay, of all of these, what's changed the most? Oh, that's so
Right.
interesting.
But if we do study, in
Yeah,
fact, most of the time it's important
yeah,
to study, to
yeah,
have
of
single
course.
sex studies as a show
Yeah,
like this, and you always say single
yeah,
sex studies. Single sex studies in this particular case can yield a completely different result.
yeah, yeah. I'm not surprised there either. Um, okay, that would be really interesting. I look forward to seeing that data because that would be something that I would predict based on some of the early, the first work that you showed. Um, so you had the one hit, like one, and then you waited 1 week, and then you had like a two hit. That wasn't the chromatin though. That was something else.
Correct. This was the chromatin. Yes.
Um, so it seemed to me I wasn't quite clear if this is true or not, so you can correct me. But did the one hit, I don't know what else to call it besides one hit, did the acute stress, I guess that's all I have to call it, did it actually change more accessibility in the females compared to the males?
I
Or
can
no?
go back if you
Yeah,
want.
go back. Yeah.
Yes.
That,
So this is one. In fact, you
yeah,
have for each line females, males, females, males, and this is one episode of acute stress one
right.
week later.
Yeah.
It
Oh, I
does,
guess it's hard to say because is it like a darker in the control males or I was
yes,
trying to get an idea of the
but it does because otherwise you wouldn't have if this heat map only shows statistical differences.
right.
So
Okay.
if you do see this type of change 1 week after, there is in fact a change in chromatin accessibility in males and in females. Or in this sense, the change is positive, which means that in both cases, chromatin accessibility is increased
Is increased.
because the color is lighter.
Okay. And then can you tell me again what's FSS?
Forced swim stress.
Oh, that's just one.
One
And
acute
that's
swim stress. This is soon after forced swim stress.
right after. Okay.
The genotype, so this chromatin accessibility is way stronger. That has a way stronger effect. This is soon after the forced swim stress. So you'd expect that there are many
Sure.
many genes that need to be expressed, and in particular, this part of gene expression. So this section of DNA is more expressed than others.
Yeah, because you can imagine like I mean, this is really interesting because ARS was a week after. Is that correct?
ARS was initially, and then
Oh,
swim stress a week after.
it was a week after. Sorry, so I got them mixed up. But you can see that there could be so many different variations that you could do. Maybe you do like a week later to see, and then do your second or third or fourth hit then to see, hey, has that primed these different regions to react even more strongly or
Yes,
less.
or less.
Yeah.
Well, in that case, if, for example, I focused on females here,
Yeah.
the fact that they did have a pre-experience of acute restraint stress,
Hmm.
this is a double hit,
Hmm. Yeah.
it have less of a chromatin opening
Yes. Yes.
if you have
Right.
all the swim stress.
Right.
Somehow this is by itself an interesting
It
part
is. It
of
is
the
because or some people might think of females perhaps being more vulnerable to or susceptible. They certainly show greater depression rates. So does that suggest that they're more susceptible to stress effects? But if
well,
you're if you're inhibiting it, maybe not. Maybe there's a resilience factor, and what might that be?
that might be due to the fact that in fact when acute restraint stress by itself, if we do compare these two, females seem to adapt to respond to one hit
Mhm.
more than males.
More. Yeah, yeah, that's what
But
I thought. I could.
so the priming in that case, if the
Is
priming is different, then the second hit may also be different. And
right.
and that's by itself, that's what I was saying. If
Yeah,
we do an only female study,
right.
it might yield
Yeah.
a different types of chromatin
Wow.
opening because they're focused on female. It's focused on
Yeah, females only. And it could be really interesting to do that based on which probably you're planning to do or thinking, or maybe this is what you're thinking about doing, but based on, ovarian hormone status, for example, because that could be really interesting as well. Like, do you see a difference depending on, life stage even?
Yeah, yes, life staging or high
Yeah.
estrogen, or low
Yeah,
estrogen.
or low estrogen, exactly. Yeah. So does anybody else have a question? Because that don't shouldn't just be me asking questions. But I'm happy to, because
I'm
I'm
happy to
me.
stay here.
Um, your core experiment, I think I went back to the slide when I took a picture of it. I think it said, did you do it so it was oral core, which is nice. Did you do it for 2 weeks? Or how long did you give oral core
I did
for?
it, no, I did it in the end for a month
Oh, a month. Okay.
because
Yeah.
the first phenotype
Yeah.
that I on
Right.
the behavioral test,
Yeah.
the LiDAR box,
It was 3 weeks later, right?
the least
Right.
because I like to distance the behaviors for at
Hmm.
least days, not
Hmm.
doing like a battery of behaviors one day after the other, because this
Hmm.
biases a bit
Hmm.
the
Sure.
outcome behavior sometimes.
Yeah.
So you wait a week to do the second behavior,
Right.
and then I well,
Okay. So it was a week or more. I'm sorry, a month of court. I was just going to ask you, would you expect to see something a little different? Because I think you found still greater emotionality in the males. Is that correct?
correct.
Yeah. Which kind of surprised me.
Correct. This may this is something that by itself means it so it calls for finding, I think, the right test to
Yeah, I think that I really appreciate what you said. Like, hey, these tests have been, designed and males and also, that's also a bias to think, oh, females are going to be more emotional. And I'm not saying that females should be
yes.
more emotional, but like in response to to stress, you might expect that. Although Robert Paul Jester's work does suggest that it's in terms of the sort of classic allostatic load in humans responses, it does seem to be higher in males than it is in female, like human males than it is in human females, which is also interesting.
Yeah, he has also interesting data on gender and
Yeah,
sexuality.
yeah, yeah,
Uh,
even better. Like really, not even better. It's all really good. So maybe that actually fits with this in terms of I know it's allostatic load versus emotionality, but it's
yes.
it's really
Might
yeah, yeah,
also in fact as a limitation that I might share with you is that
yeah.
it's possible, and I think we all should thrive to uh, be exhaustive in analyzing behavior and saying maybe there are other behavioral
Mhm,
paradigms that
mhm,
we can use to
mhm,
reveal a phenotype or a behavior that we don't see with what we've used thus far.
mhm.
And
Yeah.
it needs to be sex specific because there might some behavioral paradigm may work in males but not in females and vice versa, especially under certain conditions. So
Yeah, I'm sure many of you can appreciate this, and if you can't, if you're a trainee and don't necessarily know, well, not all of them, but many of the behavioral tasks that we have out there have been largely driven by data and experiments in males, male mice, male rats, and we do need people to pay more attention to what might work a little differently or might be a more not necessarily appropriate, but a different test in females. I think that's really an important point. And then, of course, we know Rebecca Shansky's work that hey, females, at least for acute fear conditioning, can show a very different kind of behavior. So maybe that's the other thing too. I mean, splash test and light dark box. There's not too many behaviors that are going on there. Although maybe again, that's a bias. Maybe there is. Maybe there are different kinds of that are important to look at. So.
But then remember, sorry if I interrupt,
No,
but remember this also applies to building or to building an animal model. Sometimes
yeah,
in the past they would hear something like, "Oh, this animal model works in males, so if we do this, it works in
right,
males, not in females." And they would say, "Is it a matter of the animal models that is working,
right.
or the way that we use? So this stress works in males but not in females." So sometimes was like more like not answering the right question, not asking the right question. So we
I
were
love that. I love that. What a great way to end. That is a perfect ending. I need that to be the, your scientific memoir. That's the title of your scientific.
the way, Lisa, and you give a great example. I'm leading on this. So
Oh, stop it. Stop it. Well, just once again, because we're out of time, I just want to say thank you so much to Dr. Morocco, and please join me in giving the little
thank you
emojis. Thanks so much. Thank you so much for your time and sharing your
for
expertise. It
attending.
was really, really fantastic.
Thank you everyone for attending. See you soon. Bye.
Yeah, see you soon.